Mature B-cell lymphoma is a cancer of our immune system, and it comes from white blood cells that are called B lymphocytes, also commonly known as B cells, that have matured in the bone marrow but then migrated to secondary lymphoid organs such as the spleen and the lymph nodes. There, they can be activated when we have an infection, and they will play a fundamental role in our immune response to these pathogens in order to eliminate them. That's kind of a treason from these cells because they are supposed to protect us, but can actually become cancer themselves. And because they are B cells, they can travel throughout the body and kind of have this potential to disseminate the cancer through multiple tissues, which makes them very dangerous. B cells themselves mutate their antibody genes, and the goal is to improve their affinity against the pathogen that was encountered. But at the same time, they need to rapidly proliferate, which sets the stage for the acquisition of unwanted mutations. And the second step is these cells, after mutating, they need to compete with each other based on their affinity to survive. Because the cells have to survive this very intense process, they're at a very high risk of becoming cancerous. So that's where my lab comes in, and we are trying to really understand the biology of these tumors, how they develop, what makes them resistant to treatment. Then maybe we can identify their vulnerabilities, what they depend on to survive, and start targeting these processes. We utilize precise immunological techniques that allow us to actually place B cells in competition with each other and to track these cells as they compete with each other to determine what they become and how cells that are winning the competition differ from cells that are losing the competition. We are also very much interested in understanding the metabolic regulation, because for cells to survive and proliferate as they become cancer, they need energy to fuel their growth. And so we are interested in understanding what fuels these cells and how they use their energy. So maybe we can identify metabolic vulnerabilities of these tumors. We then can take tumors and actually grow them in the lab. So we can test their response to different stimuli, the presence of other cells, the availability of different metabolic precursors, which is easier to modulate in the petri dish. And we also utilize tissues from tumors. And so we hope to identify things that they really need to thrive, not only in the lymphoid tissues, but also as they disseminate to other very different tissues throughout the body. If we are able to understand what differentiates a very aggressive, non-responsive tumor, we can potentially identify the vulnerabilities of these tumors and design improved and personalized treatments for patients who currently remain incurable. I'm hoping that with this research we are going to be able to advance biomedical research, not only to understand and better harness the capabilities of our immune system, but also to understand why cancer develops and how.